Hi, this is our team. We're the 3D printed heart vein system. My name's Ilan Fair and I'm a biomedical engineer.
My name is Joshua Harris, and I'm the electrical engineering major on this team.
My name is Dragono, and I am the bioengineering major.
And I'm Peter Gory, and I'm the mechanical engineering major. The purpose of our project is to be able to print the early stages of a heart. By printing the early stages of a heart, we can make more complex chambers and eventually print a whole heart with living cells.
This presentation consists of explaining the problem we have today regarding donor hearts and how we plan to fix it. We utilize cell culture methods, 3D bioprinters, and electric stimulus to create our device. Videos on the bioprinters and the electric stimulus are shown later on. The problem we face today is that as of October of 2021, 106,834 people who are in need of an organ transplant have already signed up for a transplant waiting list in America. Every 9 minutes, another person is added to this list. And on average, 17 people die each day waiting for an organ transplant, as the wait list is expected to take about 6 to 9 months. Of these patients, up to 50,000 of them are heart transplant candidates. Because of the severe organ scarcity, the healthcare professionals face the challenge of life and death. They have to decide who gets a heart transplant and who does not.
Manufacturing a potential heart replacement means that theoretically no one will have to wait 6 to 9 months for a donor transplant again. We want to save those lives by doing so and combining additive manufacturing technology with stem cell culture to develop a 3D bioprinted heart replacement. Cell culture is an essential process that generates a large amount of living cells, but it is expensive and time-consuming. It requires the use of specific environmental conditions, such as temperature, oxygen, humidity, and the CO2 levels. The cells also require a number of substances and materials, like cell media, that promote intercellular signaling, which is required for cell growth, differentiation, and proliferation. The chosen cell line, H9C2, is a skeletal muscle-like subclone derived from an embryonic BD1X rat heart tissue. After the cell's rate of development and proliferation slows, they are visually analyzed through microscopy, then removed from the culture dish, cleaned and separated from the cell media before they are mixed with the 3D printer's biogel.
Looking at the bioprinting process of this project, we use two different types of printers all year long. We use the BioX and the LumenX. For our material, we look to use a GELMA-based bioink. What the GELMA allows us to do, it allows us to print standalone structures and scaffolds without the use of a mold. Molds can cause damage to the material, so we try to avoid that. Then, for our GELMA, we also needed to have laminin and fibrin synthetics in this. The reason for this is because muscle cells need a collagen coating to allow them to communicate and differentiate properly to elongate. Without this, the cells just kind of stay in one place and do not properly grow. For the cell mixing process, we take the cells that have been growing in a petri dish, take those out, mix them in with our material, and then print and then cure using UV light and then incubate. Or we could plate the cells on top of the surface of our scaffold after the scaffold's been sterilized and then incubate and grow. Cells are very adhesive, so this plating method works, and then obviously the cell mixing process is more traditional.
The main function of our project is to be able to contract cardiomyocytes to control a flow rate. The heart is connected to the nervous system, and since we are fabricating our own heart, there is no possibility we could connect a mechanical heart to a nervous system. This is why we don't have to think about our heart beating. Our body does it for us. We need to replicate this bodily function. To account for this, we are working to develop an electrical stimulus system that will deliver a shock to the muscle cells as a result, produce contractions. We went about this by designing a circuit that will replicate how the nervous system sends stimulus to the heart. To prove that our circuit worked, we ran a test on an arm and checked if it would cause a contraction, and we were very successful.
For our printers, we switched to the BioX printer. Right now, it is using pressure to extrude the material as an additive printer, and from there, each layer is cured by a UV light, and then it does a final UV light curing. Next, we had the LumenX printer, which is a DLP printer. It takes a liquid and using direct UV light cures the liquid onto a build plate layer by layer. The build plate rises to get our mold and model. Now looking at the peristalsis simulation, we want to replicate a peristalsis contraction. This is the same type of contraction that's in your throat that pushes food down. Using ANSYS, we know that the flow rate increases as the diameter decreases. And so we simulated a 1 millimeter flow through the inlet, and through the contraction, the flow would increase along the directions of the contractions one by one, and we got an outlet velocity of 4.6 millimeters per second.
For electrical stimulus, we used an Arduino, a relay system, electrodes, alligator clips, and wiring to cause a contraction in the forearm. We used the Arduino to program the relay as a switch, allowing us to change when we want to supply voltage. We were able to control when electrical stimulus was supplied. Additionally, we were able to control the amount of voltage and current using the variable power supply. This proved that the same can be done for cells using additional supplies, applying and wiring, and placing electrodes in the dish containing cells. In the future, we plan to run tests on stimulating cells.
In the cell culture, the cardiomyocytes were examined visually using a digital inverted microscope along with colored lenses, and they were all found to be healthy and viable. After the cells were counted in the petri dish, it was determined that the number of cardiac cells quadrupled over the first 5 days of cell culture.
It is a major endeavor with the potential to save millions of lives and have a huge influence all across the world. We have conducted an extensive study on the project's subtopics, such as cell culture, bioprinting, biocompatibility, tissue stimulation, and so on. As a result, as we move into the production stage of our schedule, we've outlined the protocols and procedures we will follow. The protocols and techniques for tissue culture are linked to the cells and culture material used. We've also looked into and tested a variety of bioinks for use in the bioprinting process. We have had many failed prints, but from each one we have learned important characteristics and variables from managing print quality. As a result, we decided to use a GELMA-based bioink as a framework for the muscular tissue and fibrin as connective tissue between the different layers. For the electrical simulation, there have been multiple additions to better suit the project's needs, and the changes were necessary to deliver electrical simulation with controlled voltage and current. These decisions have been one of the most difficult challenges thus far. There are numerous approaches that could present a solution to our proposed problem, but we are confident that we have chosen the appropriate materials and procedures to ensure the system's operation, taking into account the facilities, resources, and budget constraints. In the following phase of the project, we will merge the separate fields, electrical, bioprinting, and cell culture, to begin the manufacturing process and build our initial proof of concept and later prototype. We would like to give thanks to the Hardiman Scholars Program for providing us the funds to start our project. Also, to Dr. Behrooz Ovasi for the initial concept for the project. We would also like to give a special thanks to the Biology Department of Fairfield University for helping us with the cell culture process. More specifically, Dr. Shelley Phelan, Elizabeth Ricci, Kenesha Norford, Cassandra Riley, and Kate Junkins for introducing us to Dr. Phelan. Thank you for watching our presentation about our project. If you have any questions, feel free to contact us on the email shown.